Hi everyone. We are going to be talking today about Chapter 14, Hypersensitivity Reactions. And so, um, we've talked about the immune system an awful lot this semester, thankfully. And when we do, we talk about it mostly in terms of it being a defense mechanism against, a foreign pathogen that's invaded a host. But we've alluded to it a couple of times that too much of a good thing is not always so good. And, we're going to see not only tonight in hypersensitivity reactions, but, as we go forward through the semester, some of the disorders of the immune system that illustrate this. Kind of too much is not always good. Hypersensitivity reactions are the first one we're going to look at, and that's tonight's lecture. Let's take a look at a couple of the key terms before we start, just so we're all using the same language. Hypersensitivity refers to really an exaggerated or inappropriate immune response to a generally harmless antigen that can cause injury to the tissue, disease, or even death. Something like pollen or a peanut can cause a overzealous immune response that can harm the host. An allergen is really, we're talking about any substance that's antigenic that gets recognized by the immune system as foreign. It's most often eaten or inhaled, and it ends up triggering what we call a type 1 or allergic reaction. Anaphylaxis is an acute allergic reaction to an antigen. You think of things like a bee sting, or eating a peanut, to which the body has become hypersensitive. Atopy is an inherited tendency to respond to a naturally occurring antigen with this really hyperactive overproduction of an immunoglobulin IgE. A couple of immunologists your book speaks about, Gell and Coombs, and Coombs is a name you're going to get to know and identify in both this class and in blood banking next semester. But these scientists classified the reactions of hypersensitivity in a couple of different ways. They started out by naming them with 4 different categories of hypersensitivity. And if we look right here, here are 3 of the classes, and there's the 4th one. Types 1, 2, 3, and 4. So they classified the hypersensitivity based on kind of how the process happened. What was the mechanism of hypersensitivity? Was it antibody mediated, or was it T cell mediated? So that was kind of our first cut. Then they further classified it by the response time. Did the response of the immune system develop within minutes, pretty immediately, or did it develop, hours and days, over a period of hours and days? So those are really our 2 factors that are going to break down our classification. So the 2 classes that we look at are the antibody mediated immediate. So it's hard to say. I keep stumbling over this. Antibody-mediated immediate forms. These develop minutes to hours after the antigen is first introduced to the host. And there are 3 of the 4 types are of this classification. So 3 of the 4 types have an antibody reaction, and their immediate response. Type 1 is the one we think of as allergies. These are immediate hypersensitivity or anaphylactic hypersensitivity reactions. The second one is the antibody-mediated cytotoxic hypersensitivity reaction. We're going to see that in just a minute, what that's referring to. Type 3 is referring to the complex-mediated hypersensitivity. Here, complex is referring to antigen-antibody complexes, not how confounding or complicated it is, but really that antigen-antibody complex. And then our only cell-mediated delayed form are T-cell mediated. So they're cell-mediated or delayed hypersensitivity is what type 4 is. Not too hard to follow along with that. Let's look at some of the key constituents of type 1 hypersensitivity. We're going to be looking at the fact that this is primarily an IgE antibody that's involved in type 1 hypersensitivity. Remember, this is the allergies, right? What do we remember about IgE? It's synthesized primarily in the lymphoid tissues of both respiratory and GI tracts. And, it's important to know that in hypersensitivity reactions, let me see how to say this. This process is regulated by T helper 2 cells. So let me back up and tell you that, under normal circumstances of an immune response, the presentation of antigens, those antigenic peptides that get digested by antigen-presenting cells and then get complexed to MHC complex 2, that MHC2 antigen structure, when it's presented by the APC, is normally presented to T helper 1 cells. And this is important because what T helper 1 cells do is they produce an interferon gamma, that's going to suppress IgE production in the presence of this particular antigen. But in people with allergies, we're going to see there's a genetic change that happens where instead of naive T cells being stimulated to T helper 1 cells, they end up as T helper 2 cells. And T helper 2 cells are produced instead. They respond to that same antigen MHC2 complex in a way that causes the production of cytokines that end up being released and causing B cells to not produce what we would normally expect after a first or second exposure to an antigen. We normally think B cells will produce IgM and IgG, but instead, under the action of these T helper cells, the cytokines the T helper cells are responsible for reproducing cause what's called a class shift. And these B cells start to produce heavy chains that are epsilon instead of either gamma or mu. So we end up with a production of IgE. And this is going to be a sustained production. This is going to be much higher concentration than we would normally see. And this IgE we're going to see is going to be responsible for those allergic reactions that we think about. So does that make sense so far? I sure hope so. If we talk about some of the cells of hypersensitivity reactions, we're talking about mast cells and basophils. We've talked about these before, so we're going to go through this quickly. Mast cells are considered the primary effector cells of the immediate immune hypersensitivity type 1 reaction. Their precursors are normally in the bone marrow. They migrate to where the tissue. This is the mast cell. The granules are going to contain a number of different mediators, but one of the primary ones that we think about is histamine. We're going to see a couple of those in just a minute. And we're going to talk about this high affinity receptor in just a second. Basophils, on the other hand, are represented in very, very small numbers in the peripheral blood, but they can leave the vasculature and go into the tissues during inflammatory responses, where they accumulate, and they're going to take place in this immune response as well. Their granules also are very histamine rich, and they also have these high affinity receptors on their membranes for IgE. What do these high affinity receptors do? The purpose of these high affinity receptors, we're going to see in just a minute that there are receptors on the cell surface of these two kinds of cells, mast cells and basophils, where IgE, the Fc portion of IgE, the epsilon chain, is going to bind right onto this cell. It's going to be a terrific way for IgE to bind to this, and that binding of IgE onto a mast cell at those receptors you'll see in a minute, they're called Fc epsilon R1 receptors. That binding of receptor and IgE is going to trigger this hypersensitivity reaction. So the fact that there's high affinity receptors that are going to bind IgE, they're going to bind on quickly and they're going to bind on tight. So we'll take a look at those in a few minutes. A couple of other things. There are we're going to be talking about some chemicals that get released from the granules of both, we're going to primarily just say mast cells and basophils in the early and later phases of hypersensitivity type 1 reactions. They're classified in two ways. They're classified either as a preformed mediator or they're classified as a newly synthesized mediator. Your book also calls the preformed mediators primary, and they call the newly synthesized mediators secondary. If you look on, I actually made a little table on another slide from now, but it's table 14-1 in your book. This is a small list of representation of the preformed and newly synthesized mediators that we're going to be talking about. I'll talk about these in just a moment, so I'm not going to go through them now, but they're classified here for us to look at. And here's where we're going to look at them. So as I said, primary or preformed mediators, when we're talking about these, we're talking about mediators or chemicals that are existing inside the cytoplasm of mast cell, or the cytoplasmic granules of mast cells and basophils. As I said, there are a number of those preformed or primary mediators: histamine, heparin, something called eosinophilic chemotactic factor of anaphylaxis, or ECFA. There's a neutrophil chemotactic factor, and then we have some enzymes, some proteases. And what are the purposes of these? We're going to talk about them in a little bit more detail, but histamine really is involved in smooth muscle contraction, vasodilation of the vasculature, specifically out in the capillaries and the tissues, and this results in some increased vascular permeability. Heparin has similar effects as histamine does, but we're going to see in a minute that it actually will also impact the T helper cells in just a few moments. Eosinophilic chemotactic factor is obviously a chemotactic agent, as are the neutrophilic chemotactic factors. Some of the proteases that we look at can be conversion of complement, factors of complement. It also can stimulate mucus production, which is a hallmark of type 1 hypersensitivity reactions. It can also activate some of those other cytokines, resulting in newly synthesized or secondary mediators. So what these secondary mediators are talking about, when we're talking about newly synthesized factors or mediators, what we're saying is that these are after the preformed mediators are released, the reactions that we get is that some of the preformed mediators tell the same basophils and mast cells to produce some more. It tells them to synthesize some new mediators. So we have these existing in the granules of the cell already, and then we can have the cell producing some additional ones. And some of the ones we're going to produce are, we are most concerned with things like the prostaglandins, which has a reaction very much like histamine, a vasodilator, increasing vascular permeability. So it increases and amplifies the immune response to this process. We can have some chemotactic agents calling additional neutrophils and eosinophils to the area. Leukotriene, you'll see that sometimes characterized or written as an LT for leukotriene. So, just like we've seen interleukins like this, leukotrienes are LT. Some of the leukotrienes also have effects to increase vascular permeability, but also, constrict the bronchial passages as well as mucus secretion. Platelet activating factor is also released as a newly synthesized mediator, and it results in platelet aggregation. And then obviously any number of cytokines are also produced by mast cells and basophils that have an impact in increasing inflammation and increasing immunoglobulin IgE. So let's get down to the brass tacks when we're talking about hypersensitivity, type 1 hypersensitivity reaction. We also know this is anaphylactic hypersensitivity or immediate hypersensitivity. Hypersensitivity reactions are characterized by having really a excessively large IgE production relative to a small concentration of allergen that was presented. So it's really an exaggerated response. We're going to see in the type 1 hypersensitivity reactions that it's a cell-bound antibody that reacts with antigen to release those mediators from either mast cells or basophils primarily. Other immunologic cells can be involved, but let's, for the sake of argument, now talk about mast cells and basophils. Some of the key points about type 1 hypersensitivity reactions, it has that short lag time. If you remember, we've used that term before when we're talking about the amount of time for an immunogen to be produced. Not an immunogen, I apologize, for an immunoglobulin to be produced. We saw it when we looked at the primary and secondary responses of antibody production. That short lag time after the host was exposed to an allergen is a hallmark of type 1 hypersensitivity. Remember, we said it was an immediate response, right? So it's got a short lag time for that reaction of antigen-antibody binding. Those key components we just went through, so I won't go through those again, but we're going to see there are 3 phases of the immune response, and we're going to go through those in just a minute. So remember that this is, an allergy reaction. Allergens, those are the types of antigens that are going to induce type 1 hypersensitivity reactions. And I gave you some examples, and we'll see some more in a few minutes. Things like, pollen or peanuts or something like that. Oh, yeah. So, as I said, how did I go past? Where did I go? Where did I go? Where did I go? Up, 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 up. I apologize. 3 phases of the immune response of hypersensitivity. So let's start with sensitization first. That term should sound familiar to you. We talked about it in terms of antigen-antibody binding before, and that's exactly what we're looking at here. So in this case, in the sensitization phase, once the host is initially exposed to an allergen, the antigen-presenting cells, just like we learned way back when, the allergen is brought into the antigen-presenting cell where it gets processed into small little peptides. The peptides bind to our friend the MHC class II complex, and it's brought to the surface of the antigen-presenting cell where it gets presented. Remember, in type 1 hypersensitivity, gets presented not to type 1 but to type 2 T cells. Remember, under normal circumstances, that would be a T helper cell 1, but the genetic predisposition that we're going to see in a few minutes is that T helper cell 2s are going to be the predominant T cell. And this is important in this process because the T helper 2 cell will release specific cytokines, interleukins, a couple of them, interleukin 13, interleukin 4, which work, synergistically to have B cells, so that B cells are going to be induced to produce not IgG, not IgM, but rather IgE instead. So that's that class switching. So under the action of these cytokines, B cells are going to switch on the production of epsilon heavy chains in order to produce IgE antibodies. The B cells are differentiated, as I said, to produce IgE antibodies. This is supposed to be a plasma cell at this point. And IgE antibodies are released. IgE, the Fc portion, remember this is the Fc portion, that's the FAB portion up here. The Fc portion of the heavy chain down here is going to bind to those receptors that I talked about that are on mast cells and basophils. So that receptor is the Fc epsilon R1 receptor. And so this receptor binds to the Fc portion of the IgE antibody. And this binding, once the antibody is bound to the cell, that's called sensitization, and that's the end of the sensitization phase. IgE bound to the Fc epsilon R1 receptor ends sensitization. The binding there results in a couple of things. It will bring more basophils to the tissue, and it's important to note that basophils and mast cells have a huge number of receptors on each cell, much more than anything else. And so they have roughly 200, up to 200,000 of those Fc epsilon R1 receptors. And that once we bind that IgE to the receptor, the lifespan of IgE increases from almost triples in length and time. So this process, we have not only produced more IgE here, we've turned on B cells to produce it. Plasma cells produce it, they bind off. So not only have we produced a lot of IgE, but we've increased the longevity of the effectiveness of IgE. So that's how, I think if you take those two things into consideration, you'll see why all of a sudden we're starting to set ourselves up for kind of the perfect storm of a hypersensitivity reaction, of a prolonged hypersensitivity reaction. As I said, this sensitization ends the sensitization phase, not ironically, and we move into the activation phase. The activation phase represents really the encounter of these mast cells, let's say it's a mast cell, with another molecule of antigen. So at this point I've got IgE bound to a whole bunch of those receptors. Remember, we have 200,000 of those receptors on there. So we're pretty likely that the antigen is going to be able to bind to the IgE the way we need it to for this. So what happens in the activation phase? A bivalent or a multivalent allergen. Remember we talked a few weeks about valency, and bivalent and multivalent just means that there are multiple binding sites, multiple epitopes on the allergen, right? At least 2 epitopes, and multi is many epitopes. So we have multiple epitopes on the allergen. And that's going to be important because we need that allergen, that little teeny allergen that we saw here and here, needs to bind to at least 2 IgE molecules that are in close proximity next to each other, that are bound to the cell. And they need to do that so that we create what's called a cross-linking. So we're going to link immunoglobulin, allergen, immunoglobulin. We've linked that up. That cross-linking has an important purpose. Once that cross-linking takes place where the allergen is linked to a couple of different IgEs, it triggers this cell to drop the contents of its cytoplasmic granules. The granules, those are the ones that had those primary mediators or the preformed mediators, the histamine, the heparin, the eosinophilic and neutrophilic chemotactic factors, what else was in there? The proteases. So those have all been released from the basophil or mast cell, resulting in allergic symptoms. The smooth muscle contraction, the vasodilation. We're going to see that those are going to have an effect in causing swelling and redness at the site. So this release of those primary mediators is what happens during the activation phase. And the activation occurs, activation to release those cytoplasmic granules, what am I saying? The granules with the contents of the granules within the cytoplasm. That cross-linking activates the, let's say, mast cell or basophil to release the contents of their granules. And it's done in this because of this cross-linking. I told you that part of one of the effects of these granules is to induce what? It's to induce the synthesis of some secondary mediators. So those granules that got released here are going to signal these cells to produce some additional mediators. And they get released 6 or 8 hours after exposure to the allergen. What kinds of things are we looking at here? We're looking at mediators that are going to do things like call more cells to the area. They're going to secrete additional chemicals on their own. They're going to cause allergic reaction. So let's look at these. We looked at the four that are listed here before: the platelet activating factor, prostaglandins, leukotrienes, and cytokines. What's important about those that we haven't already discussed? One of the cytokines that gets released, interleukin 5, is going to actually trigger the bone marrow to produce and release more eosinophils. And that will allow, once eosinophils move to the area, they get produced, they get released from the bone marrow, and they go to the site of the allergens into the tissues. They're able to enhance this reaction by releasing their granules, and they have a very pro-inflammatory granular content within them. So one of the actions of these secondary mediators is to increase some of these other cell types and produce more of them. Some of these mediators induce cells to leave the circulation and enter the tissue, and those cells again are going to release more mediators. So it's really kind of enhancing and continuing to prolong this immune response. Let's see. Yeah, I think that's it. So question 1, what actions result in the degranulation of basophils and mast cells? If we looked at these, I think it would be fair to say that the answer is C, cross-linking of a bivalent and multivalent antigen. I said earlier pretty much anything can cause a type 1 hypersensitivity reaction, but here's a bunch of the most common ones. Bunch, that's the technical term. Pollen, mold, animal dander, dust mites, venoms, certain foods, peanuts, shellfish, allergy. Wheat is another one we see. Some drugs, penicillin. A lot of people are allergic to penicillin because it was a little hapten that would bind to molecules and make it antigenic. And latex, that's something we worry about an awful lot in healthcare. And in fact, at this point, it's such a risky allergen that most things are latex free, as you know. It's important to look at the genetic and environmental influences of type 1 hypersensitivity, because really this is understanding the environmental and genetic components of allergic diseases is becoming kind of a public health issue. It comes from the fact that about 30% of the population suffers from some kind of an allergy, many of which are associated with the development of asthma. And so that's a public health risk that needs to be addressed. While we know that there's genetic influence, we also know that in the West that some of it is compounded by our lifestyle, including things like spending a lot of time indoors, diet, air pollution, psychological stress. So combining looking at genetic and environmental influences, kind of the epigenetics of health, is really important to what we do in understanding type 1 hypersensitivity and being able to, treat the patients that are developing these allergies. So as I said, 25 to 30% of the population has some kind of allergy. And there's really strong correlation of genetic influence, and we can see it when we look at family structures. Families where one parent has an atopic or allergenic disease, at least a third of kids almost will also be allergenic. If both patients are atopic or have the allergy, at least half of their kids will have some similar or if not worse, allergies. So you can see that there's a really strong genetic correlation between atopic disease in the parents and the offspring. However, I think everyone's heard the expression that correlation is not causation, and I think we're seeing that here. Genetic predisposition doesn't eliminate environmental influence, because there's clearly some environmental influence. Things we couldn't discount if we're looking at families is that family clustering of disease is not just genetic. Maybe it's because they share the same environmental exposures. They live in the same house. They are exposed to the same toxic substances or air quality. And so they've also done studies, and if you look at in the references, the reference that I used, I think you probably can trace it back to some of the identical twin studies that were done, where they found that identical twins that were raised in different environments still had similar disease states when they looked at rates of allergy and asthma. So the genetic influence is quite real. If we look at some of the specifics of genetics and the environment related to this, let me just fix this one thing. I noticed it earlier and I forgot to fix it. Which I'm not going to spend a huge amount of time doing it right now. Oops, of course it's not going to do it because I'm talking. I'm sorry, I didn't mean to do this here, but there we go. let's see, slideshow, sorry. Okay, it wasn't going to make sense otherwise. So we're looking at the specifics of genetic environment and environment related to type 1 hypersensitivity. We're still on type 1. Yes, it's true. Several hundred genes have been identified relative to type 1 hypersensitivity. 4 basic categories of the impact or the influence of the genes that are related to hypersensitivity. Number one are the genetic polymorphisms. Do you remember the definition for polymorphism? It's referring to genetic mutations found in at least 2% of the population. So we're talking about the first category are those genetic mutations that influence or alter the effectiveness of physical barriers within the innate immune system, those that are able to keep allergens out. So maybe someone has reduced capacity of mucus production, preventing inhalation of something, or something within the GI tract, for example, that maybe isn't an as effective physical barrier as someone else. So there's some kind of genetic mutation that influences or alters the effectiveness of their physical barriers. The second category is really talking about the antigen recognition coding for pathogen recognition receptors and toll-like receptors. And I think that antigen recognition process that we talked about earlier this semester is such an important thing, and if it's not effective or it's altered in some way, it can influence the overproduction and overstimulation of the immune response. The third of the 4 basic categories can be classified as dysregulation of the adaptive immune system, including cytokine production, that T helper cell differentiation, so that instead of T helper 1 differentiated from those naive T cells when an allergen is presented, instead it's triggered to be a T helper 2 that will accept antigen presentation. Also some, that HLA, or what is another term for that, the MHC coding for antigen presentation. We talked a lot earlier this year about MHCs and the genetic influence on being able to effectively present an antigen and how that impacts immunogenicity of something foreign. And finally, there can be dysregulation that we saw, that we've talked about briefly already, about that hyper IgE production that can result from this during type 1 hypersensitivity. So those are 3 of the 4 categories. And the fourth one is mutations in the genetic codes that are going to control the inflammatory response. So we, just as a general note, I said there were hundreds of genes that were identified, but the tendency to respond in this way, in a hypersensitive way, is linked to several of the major histocompatibility genes, the HLA, DR2, DR4, and DR7, if we remember. Those were on the loci of the chromosome. These ones, these HLA molecules here that are linked to the MHC genes, are more likely to trigger that sensitization that we just saw of IgE sensitizing and binding to mast cells and basophils at that Fc epsilon R1 receptor. But as I said, we're not going to pretend that environment doesn't have an impact to this. In addition to some of the things we talked about, one of the environmental exposures that can influence this process is prior exposure to infectious organisms. We have seen study after study that was looking at what's called a farm effect, and that is that either in utero or in early life exposure, people that were exposed to antigens at a higher rate were induced to develop not Th2, not T helper 2 regulatory cells, but they were induced or more likely to develop those normal T helper 1 cells that will suppress the overproduction that we saw with T helper 2 cells. So it's a really important process, that an example of an environmental control that we can point to. What are some of the clinical manifestations of hypersensitivity reactions? We can talk about, let's see, what are we going to talk about here? I don't know, I'm losing track. We saw that the clinical manifestations of allergic reactions result from the release of mediators. But we can also see that the severity of the reaction of the clinical manifestations can be based on a number of different things. It can be based first on how was the host exposed to the allergen. Was it, if it was inhaled, they're going to have some respiratory issues. If it was ingested, we're going to see the clinical manifestations in the form of a GI issue. If it was injected, we're going to see those clinical symptoms or manifestations appearing as kind of an all-over systemic response because it was injected into them. So it's going to travel through the system, through the vasculature quickly. What else can influence the clinical manifestations or appearance of what we see? Dosage and frequency. You know, if you are exposed to more of an allergen, you have a higher response. If you're exposed to it more frequently, what do we know about our immune system? Boy, does it like a good memory, right? And we built some memory cells when we were activating those B cells to produce IgE. We're also producing some memory cells. So frequency of exposure just makes our response faster and better, right? So these two things can influence the severity in great detail. What are some of the clinical things that we see, the clinical manifestations of type 1 hypersensitivity? Anaphylaxis, we've already talked about, which was an acute allergic reaction. Rhinitis is just hay fever. That's really, kind of that annoying, sneezy, runny nose, watery eyes kind of a thing. The problem with rhinitis is that it sounds kind of benign, but if those conditions kind of reach the lower respiratory tract, you end up with asthma. And asthma is really just, really breathlessness. Some of the manifestations we see from food allergies, things like to cow's milk or peanuts, eggs, wheat, are GI symptoms. So I guess I probably should have put GI symptoms from food allergies rather than vice versa. Some of the symptoms we see: cramping, vomiting, diarrhea. One of the clinical manifestations we see of some type 1 hypersensitivity reactions are dermatologic. They're inflammation of the skin. What do we see in those cases? In the picture that you see to the right is a picture of inflammation of the skin. What we see in type 1 hypersensitivity reactions related to the skin is what we call a wheal and flare reaction. Wheal and flare reaction. And a wheal and flare reaction really is just talking about the wheal is swelling, and it's caused by serum moving into the tissues, or plasma, I should say, liquids, body fluids moving into the tissues. That's called the wheal. So you can see this is very swollen here. And the flare is the red. It's the erythema, the redness. Erythema is just talking about redness. And this whole thing the wheal and flare together comes under the general heading of urticaria, which is just the hives. It's just hives. Some of the other skin manifestations we see, eczema is another one. So I think eczema should have been cleaned in a little. And then systematic anaphylaxis, what do we see? We see, oops, I'm just going to fix this so that you can have it on your version. Systemic anaphylaxis is the most severe because it really can travel through your body systematically, potentially fatal. What are some of the things we see? Laryngeal edema, swelling of the throat, closing of the airways, right? Skin manifestations that we saw, some vascular congestion, urticaria, angioedema, swelling around the heart, and really systemic shock. In general, though, in all of these cases, the severity of the reaction is going to be dependent on those things that we talked about before: route of exposure, dosage, frequency. So it's important. Because of these kinds of a thing, this is why when we talked about latex being really problematic in healthcare, we were exposing people to latex constantly in very high doses, and it was problematic. How do we treat hypersensitivity reactions? Well, let's start with just avoiding them. It's always a good thing. If you know you're allergic to something, keep away from them. So first, avoid your allergens, and if that doesn't work and you have a reaction, there are other things we can do. There are various drug therapies. The ones that we tend to think about are things like an EpiPen that contain epinephrine, which acts as a vasoconstrictor, keeps the allergen from kind of going systemic during an anaphylactic response. What else can we do? You've seen people take antihistamines, bronchodilators, things that will keep your airways open. We have things that are called mast cell stabilizers, keep them from overproducing. Corticosteroids, which reduce the overall immune response, the inflammatory response. And I repeated epinephrine there. There are some more specific treatments if we need them. There are monoclonal anti-IgE antibodies like omelisumab. Let me see if I can say it again. Omelisumab, which binds to, this is an interesting anti-IgE antibody in that it binds the Fc portion of IgE that would normally bind to the Fc epsilon R1 receptor. So if you have the little Y, right, it's binding to the Fc portion of that. So there's a monoclonal antibody against the Fc portion that would normally bind into that receptor on the mast cell. If you are blocking that IgE mast cell sensitization, the hypersensitivity reaction doesn't take place. So that's a very clever treatment, and that's a marketable drug. But there are also things like allergy immunotherapy, or AIT, where we gradually administer small doses in increasing amounts over a period of time, like a year or more. We gradually increase doses of allergens that we administer to patients. This is a process they call hyposensitization. And what it's designed to do is to have the patient develop IgG response so that and build up IgG memory, build up memory of production of IgG. It's designed so that when the patient is exposed to the antigen later in life, that their body will respond by producing IgG instead of IgE. And what happens is if we can have IgG be a primary responder, then the immune response is going to eventually develop where the MHC2 class antigen presentation will be to T helper 1 cells instead of those T helper 2s that would trigger a hypersensitive immune response. So really this is designed, this AIT is really just designed to have the host start to develop to produce IgG instead of hypersensitivity response. There are a couple of ways we can test for hypersensitivity reactions, either in vivo or in vitro. There's a couple of them for each, so let's go through each. The in vivo skin tests, I think you're probably all familiar with. There's either percutaneous, which are little pricks through the skin, or intradermal. If you think of like, if you've ever had a TB test, which I know you have, that's an intradermal, between the layers kind of a test. The percutaneous testing is performed on the back or the forearm of the patient. This picture to your right is clearly on someone's back. They apply a panel of allergens to separate sites on the skin a couple of centimeters apart. They wait 15 or 20 minutes and they look for a reaction. It's a little old school, but it really works. It's a very inexpensive test, really sensitive for the allergens they're being tested for, and can be done in a doctor's lab without anything special on hand other than, you know, an EpiPen, I'm sure, and a good eye. So you wait 15 to 20 minutes, you look for that wheel and flare. So you can see there's the little swelling and there's the redness of the wheel and flare. You look for that reaction, you measure it, 3 centimeters or so is kind of typically positive, and you're off to the races. Intradermal is done very similarly, but it's done on the inner or upper forearm, and I'll tell you why in just a minute. We're going to inject a very dilute allergen, probably 100 to 1,000 times more dilute than the percutaneous. And the reason we do that is because we're injecting it into under the skin, and it's going to go right into the vasculature. So we put a very dilute allergen in there so we don't kill anyone. We're going to apply those same, a bunch of allergens on the forearm, and look for, wait 15 or 20 minutes, look for the wheel and flare. Intradermal is more sensitive than percutaneous, but it's riskier because, that pesky anaphylaxis. As I said, in both cases, after 15, 20 minutes, you look for the wheel and flare at the site of infection. 3 millimeter wheel is considered a positive test. You have to remember, like anything in the lab, that we have to do a positive and a negative control. So the negative control, we're going to inject something that isn't going to cause a hypersensitivity reaction. And we use saline. We use just isotonic saline. It's not going to cause any reaction because it's, we all have salt water in our blood. And the positive control is something that's going to cause a hypersensitivity reaction, and we think of histamine. Well, that's one of our primary mediators, so I'm sure that that's going to be a good producer of an immune response, right? So we have a positive and negative control. If those don't work, the test is invalid, sadly, for the person getting it. We have a couple of in vitro skin tests. So, whoops, I said that before. In vitro tests. Sorry, I think I did it on a bunch of these, so I'll correct it now, and I'll correct it from that point forward. A couple of in vitro tests we're going to look at. In vitro tests that we're going to be looking at, we're going to look at 2 types. We're going to look at ones that are allergen-specific IgE testing. So we are going to be looking for the IgE to a specific allergen. You got to know what you're looking for here. Otherwise, it's a little bit of a shot in the dark. But this is it's a very good test. It's more expensive than we just saw with the skin tests. However, it's safer. We don't have any risk of producing anaphylaxis. And a lot of people, especially young children, apprehensive adults, don't really want to get multiple skin pricks or intradermal injections. But as I said, you got to know what you're looking for. So let's look at the test method itself. This is an automated, non-competitive fluoroimmunoassay, where if you follow along with the picture here, and this picture is from your book, we have a test well with antigen on it. This is the specific antigen we're testing to see if the patient has been exposed to. We draw the patient's blood, we add in patient's serum in here. Remember, in our book, red is indicative of the patient. So we have patient antibody and seeing if it matches this specific antigen. We incubate them, we let the binding happen. What do we do next? We're always going to wash between this and the next step. Then we're going to add in some labeled anti-IgE. It can be enzyme labeled. In this case, this one happened to be fluorescently labeled. This is enzyme because technically we can end up with fluorescence, colorimetric, or chemiluminescent testing with this. So it can be enzyme labeled, it can be fluorescently labeled, whichever you need. After we add the next, so we've added an antibody, a tagged antibody to the patient IgE. So it's an anti-IgE. We wash again, and after they've bound, we wash again. You incubate, they bind up together, and you end up with a complex of what? Known antigen, patient antibody, tagged anti-IgE. We washed it again. We checked for fluorescence. Because this is a non-competitive fluoroimmunoassay, the fluorescence is proportional to the amount of the allergen-specific IgE. We can measure and report in what's called international units a positive test. We're going to measure it against what? A standard curve, right? We will have created a standard curve of fluorescence. We measure the fluorescence and measure down to the concentration, and we can tell what the concentration of the patient IgE is. Simple, easy peasy. We know all about this kind of testing now, right? Right. This is old hat for us after last week. The next one, the next in vitro test is for total IgE. I am going to apologize. This test, this allergen-specific test, is formally called the radioallergosorbent test, or the RAST test. When I was a young buck, they called it a RAST test. The total IgE is the radioimmunosorbent test, or the RIST test. RIST. So if you hear that term, sometimes they get used interchangeably still, even though they're not technically the same. Well, because this used to be radioactive, for example. We don't do that anymore. So what's the purpose of IgE, the total IgE? Well, when we just did the specific allergen testing, we were looking for a specific allergen, right? We were looking to see if the patient had an IgE developed against a specific antigen, indicating they had been exposed to that allergen. In total IgE, we're really looking at kind of monitoring things. We're looking to see if they've been exposed to something that caused an increased IgE, but not necessarily antigenically monitored. Maybe, helminth infections, those are worms, right? Other parasites. Some disease states like Wiskott-Aldrich or DeGeorge syndrome. Sometimes we can look at things and we can monitor things that we could be looking for the presence of if there's some kind of immunodeficiency, which Wiskott-Aldrich is an immunodeficiency syndrome. We may want to be looking at something that just if someone had an exposure to it before, an exposure to something that caused an increase in total IgE. Sometimes they use it for monitoring allergy therapy. So how do we do this total IgE test? This is really very simple. So in this case, because we're looking to see what the patient's total IgE is, we're going to have an anti-IgE that's prepared and bound to the membrane, because we're looking for IgE. So I'm going to have an anti-IgE reagent that's bound in. I'm going to do what? I'm going to draw the patient's blood. I'm going to put the serum in, and there's the patient's IgE. This is an anti-IgE. It binds to one of the receptors or one of the epitopes on the IgE molecule, and they bind together. And they create an anti-IgE, IgE complex. What do? We rinse. Rinse away any excess patient IgE. Then we're going to add in a reagent tagged anti-IgE, a second anti-IgE, and it's going to bind to a different epitope than this one does. It's going to bind to a second epitope on this patient IgE. We're going to let that incubate and see if we form a complex of antibody, 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 right? Antibody, patient antibody, tagged antibody. Going to wash again. We're going to look for fluorescence. The fluorescence, again, this is not a competitive test. This is a non-competitive test. I don't know if I put it here. How do we know it's not competitive? Because I don't have two free solutions of antibody here. I have a bound one, right? Everything is binding independently. The patient is binding independently to this. There's no competition here. It's proportional to the amount of immunoglobulin that the patient had. As I said, total IgE is helpful in monitoring things. It's not going to necessarily correlate to current clinical allergies. The test is could be elevated in something where maybe you have a chronic condition like helminth infections, where they don't I don't think everyone's going to have nightmares tomorrow when you think about having a tapeworm or something, that you're in a constant state of response with IgE. And in certain immunodeficiencies, as I said. So it's helpful. It's a helpful test, but it's got a specific purpose to it. So question 2, the allergy-specific IgE and total IgE test can both be classified as what? Well, we know they're both non-competitive, but what are they, homogeneous or heterogeneous? Well, they require a wash step, so they are heterogeneous. Oh, we're finally moving on to type 2 hypersensitivity reactions. Type 2 hypersensitivity are also known as cytotoxic hypersensitivity. These can also be called antibody-mediated cytotoxic hypersensitivity, which is 12 and a half. That's a mouthful. What do we know about this? The type 2 hypersensitivity reactions are one where IgM and IgG are bound to some sort of a cell surface antigen. It doesn't have to be both. It could be or. IgG and or IgM are directed against a cell surface antigen. What kind of antigens are they? We're going to have either heteroantigens, which we remember are antigens of another species that are able to produce an immune response in humans. But we can also have some things like altered self-antigens, and we'll talk about this in a few minutes. In type 2 hypersensitivity reactions, this is a cytotoxic reaction, but it's premised on the fact that there's been an antigen-antibody reaction. So, how does this work? The antibody binds to the antigen on the cell surface, and it results in cell destruction. There are a couple of different ways. The first is by the activation of complement and lysis by the membrane attack unit complex and the MAC. We know that both IgM and to a lesser degree IgG can activate complement, right? So that can cause cell destruction. The second way this antigen-antibody binding can destroy the cells and create a cytotoxic response is opsonization leading to phagocytosis. That binding brings phagocytes to the area and they phagocytize the cell with the complex on it and kill the cell. Additionally, if we have, we can have something called put each word by itself and it will sound okay. Cellular cytotoxicity, right? So toxicity to the cell by antibody-dependent cellular cytotoxicity. That's what we just said this was, right? It's antibody-mediated cytotoxic hypersensitivity. It's just a really big mouthful for saying that you can have immunoglobulins binding to the Fc receptors that are found on macrophages and natural killer cells that cause those cells to release their cytotoxic enzymes and kill the cell with the immunoglobulin. So it really is just it's a process of forcing macrophages and natural killer cells to release their cytotoxic enzymes, and it's a result of antigen-antibody binding. We're going to see this in a few minutes when we look at these reactions: transfusion reactions, hemolytic disease of the newborn, and autoimmune hemolytic anemia. So this is the first mechanism of cytotoxicity, cell destruction. There are two other mechanisms of cytotoxicity as a part of this type 2 hypersensitivity reactions. And one is one where we will inhibit cell function. And in these, we're going to see an example where an antibody will bind to a receptor on a cell and prevent the normal constituent, they call it a ligand, the thing that would normally bind to the receptor. The antibody prevents that ligand and receptor binding to take place. So it's an inhibition reaction, causes that cell not to function properly. The other thing that this kind of type 2 hypersensitivity reaction can do can increase cell function. What happens in this is that the antibody binds to a cell, usually again in a way that it's going to bind to a receptor that's going to cause that cell to produce and overproduce whatever its product is. So in the example that we're going to see that an antibody can bind to a receptor on a thyroid cell and cause the overproduction of the hormone TSH. So we're going to see those two examples in a few minutes. So those are the other mechanisms of type 2 hypersensitivity. But let's look at the ones that are the easiest, the cell destruction. Well, I say they're the easiest, but hopefully they are, because this is something we're super familiar with. So, as I said, these are cell destruction. This is we're gonna destroy the cells that have the antibody binding to them. So let's look. This is this is a cell surface antigen we're talking about. So we have a cell, in this case it's a red blood cell, and we have an antibody that binds directly to it. So this one happens to be an IgG, clearly, but it could be an IgM just as well. So we have some form of an antibody binding to a cell surface antigen. This antigen can either be a self-antigen that's been altered slightly, or it can even be a heteroantigen. It can be a different kind of a cell that has caused us to have this reaction, or we've produced an antibody to a heteroantigen that now binds to our own cell. So what happens here? If we look here, there's two distinct paths that are illustrated by this antigen-antibody binding of type 2 hypersensitivity. The first is opsonization. So we have the cell with the antibody bound to it. The Fc region of the antibody binds to the receptor on what? This is representing what to you? I think that would look like an antigen presenting cell to me, right? Or a phagocytic cell. So we have a phagocytic cell with a receptor on it. The Fc region of this complexed antigen antibody binds to it, and that's an opsonin, as we remember, and it results in this phagocytic cell engulfing this and destroying this cell, right? That's a bad thing for us. It's okay on a small scale, but when it's overproduced, like in hypersensitivity, we're going to see this is going to be problematic. What's the second mechanism of cell destruction that we've talked about? The activation of complement and lysis by the membrane attack unit. Again, we have an IgG with a red blood cell. Again, this could be an IgM, which would probably be preferable as a activator of complement, but IgG works as well. Both of them fix complement. So we have IgG, we have this lovely complement that binds into two molecules of IgG and begins the complement cascade. And we know it ultimately ends up with what? It ends up with lysis of this cell via the membrane attack complex, resulting in cell death. So check and check. That's number 1 and number 2. They didn't have a picture of it in the book. When we look at the cellular cytotoxicity by antibody-dependent cellular cytotoxicity, which we call the ADCC, but let's talk about what it is. In this, as I said, the immunoglobulin binds to the Fc receptors on macrophages and natural killer cells. So if you imagine, this would be the same thing. Here's the antigen, here's the antibody, binds to a receptor on either a macrophage or a natural killer cell, causing these cells to reduce or release their cytotoxic enzymes that are going to kill this cell. Then the phagocytic cell comes back and mops it up and cleans up, right? We're going to see these reactions here. And then finally, I told you we were going to talk about the kind of inhibition and enhancement of cell function as a mechanism of hypersensitivity. So in the example that we see in the first one, in inhibition of cell function, this is reflecting this patient over here. There's a disease called myasthenia gravis, and I don't know if you've been exposed to this disease yet, but what happens is in patients with myasthenia gravis, they produce an antibody that binds to the receptors on muscles. And those receptors are normally reserved for the ligand called acetylcholine. And what normally happens with muscle cells is that when acetylcholine binds into its appropriate receptor on the muscle cell, it causes the muscle to contract or to move. Because in myasthenia gravis patients, antibodies bind to the receptor, they block the receptor so that acetylcholine can't come in and bind where it's supposed to. And as a result, muscle contractions and movement are reduced. And you see people with these kind of drooping eyelids. They get slack musculature because they're not able to contract and move their muscles appropriately. And it's based on the fact that antibody binding to the receptor on the muscle inhibits the cell's function. Simple, right? There's another disease which sounds remarkably similar, but it's not even close. This is called Graves' disease. This is a little complex, so follow along with me. In Graves' disease, a patient produces an antibody to a receptor on thyroid cells. And this receptor is for a hormone that is produced also by thyroid cells called TSH. So antibody binds to the TSH, thyroid stimulating hormone, on thyroid cells. Antibody binds to the TSH receptor on thyroid cells. this interferes with a feedback mechanism where at certain levels of thyroid stimulating hormone, the thyroid cell slows down and scales back the production of that hormone. So when you bind off the receptor on the thyroid cells, it blocks the feedback loop, and the thyroid cells just keep producing the hormone TSH. And you end up with hyperthyroidism. You overproduce thyroid hormones. So in type 2, the binding of the antibody in Graves' disease increases the thyroid's production of hormones. So it actually increases cell function, which is also bad. So we learned about what is type 2. We learned about how does it function and how does it impact hypersensitivity. Let's look at some examples of it. We're going to look at transfusion reactions, hemolytic disease of the newborn, autoimmune hemolytic anemia. And we're going to go over these when we see them, so I don't think we need to go over it now. We're not going to really go over Goodpasture's and Hashimoto's. We'll talk about them later, and you'll do these in hematology as well. Although, anyway, Goodpasture syndrome is it's an organ-specific antibody that binds to the basement membrane of the alveolar membranes. And Hashimoto's is an antithyroid antibody that actually can destroy the thyroid gland. So that's how those two diseases are related to type 2 hypersensitivity. But let's instead spend a little bit of time looking at, first up, transfusion reactions. Transfusion reactions, we're talking about the cell destruction resulting from transfusion of whole blood or one of its components, especially like red blood cells. So we're going to look at adverse events associated with transfusion of blood or blood products. You know, we have to go back and think about your intro class, and I think we all understand the concept of blood type, blood group antigens, A, B, and O, and that type A blood cells have an A antigen on them, and type B blood cells have a B antigen on them. They're unique antigenic identifiers that will identify a patient as a certain blood type. O has neither A nor B antigens on its surface. And as we know, we have we don't produce antibodies to our own, to self. So we don't have, I'll use an example, I'm a type B, I don't have any anti-B antibodies in my blood. If I would, I wouldn't be here. We would have no blood cells left and would all be dead. So we don't produce antibodies to our own antigens, but we do produce antibodies to the antigens that we don't possess. And these antibodies are called isohemagglutinins. They're naturally occurring antibodies in the blood that are most likely produced by contact with microorganisms that have an identical antigenic determinant or epitope as humans. So as I said, we don't make antibody to our own red blood cells, but we do make antibodies to other blood groups that are not like us. So as I said, a type A person has anti-B antibody in their blood. A type O person has type A, anti-A and anti-B antibody in their blood. And a type B person, B blood person, has anti-A antibody in their blood. You're with me so far? Because those antigen-antibody reactions are really important in transfusion reactions. Transfusion reactions occur, as I said, when a patient is given blood so if you're given blood that contains antigens that you have an antibody to presently, there's gonna be an antigen antibody reaction, and the cells are going to be destroyed. So again, if I am a type B person, I have anti-A in my blood, and I'm given A blood cells, I have anti-A, and I've just been transfused with type A blood cells, I'm definitely going to have a transfusion reaction. Antigens and antibodies are going to bind, and it's going to be anywhere from minor reactions to life-threatening. The ones that are life-threatening are the ones, again, based on how much is given to people before they detect an error. But ABO incompatibility is life-threatening. There are other minor antigens that people have, or antibodies that you develop over time, that if you were given a minor antigen on a red cell, you may not have a major transfusion reaction. But we're going to test to make sure that we don't do that. So anyway, I say all this leading up to the point that in transfusion reactions, the reactions can be severe or minor. Reactions that occur kind of during the transfusion can be called acute transfusion reactions. And if they don't come up for days or weeks until days or weeks later, they're called delayed transfusion reactions. The antibody-antigen transfusion reactions that I've been speaking about, you have the antibody, you're given the antigen on a red blood cell, or vice versa, is, generally a lab error, and it's a mismatch or incompatibility of the transfused product. You can't give matching antibodies or antigens to someone who has the alternate. I told you about the isohemagglutinins already, so I won't go through that again. These antibodies, these anti-A and anti-B, really account for a tremendous number of the transfusion reactions that we see in the blood bank. And as I said, it can be severe, it can be minor to life-threatening. So you can have, do I have it on here? Yeah, so let's go through some of these. So if you have, this transfusion reaction table came from this source, and I just adapted it a little bit for us, but it's primarily from this source. If you were interested, it's in the back of the references. Transfusion reactions can be immune or non-immune related. I'm interested in the immune related ones here. When you go through blood bank, you'll do the non-immune ones, but I'm not really worried about that today because we're doing immunology. So our immune responses of transfusion reactions, we're looking at things, as I said, from a mild allergic reaction. Maybe there was a little bit of a foreign protein in a blood donor product. It could be the anticoagulant, for example. The anticoagulant that keeps the donor red blood cells from clotting. And you'll see people get, when that happens, they get a little tingling in their tongue. They start to feel it, and they stop the transfusion. The patient will be fine. Anaphylactic is similar to the mild allergic reaction, but it's sometimes more severe. We see this sometimes in a patient that has an IgA deficiency, who makes those alloantibodies against IgA through isohemagglutinins, and then gets blood products, and there can always be a little bit of immunoglobulin even inside of washed red cells. You can't get rid of everything, and so if you're given, IgA and you have an alloantibody against IgA, then you're going to have anaphylactic response. The next kind of acute transfusion reaction are these febrile non-hemolytic. This isn't directed against red blood cells, but instead what happens is that Again, even if you're getting red blood cells, a unit of red blood cells, there's always going to be some white blood cells stuck in there. And white blood cells from someone else, when they're transfused into you, they see a bunch of stuff that they don't recognize, and they start releasing their cytokines and going a little crazy, and you get a non-hemolytic immune response. It's going to produce a kind of a febrile, immunologic based reaction, trying to see what kind of interest it can drum up, for being someplace those white blood cells are someplace they don't expect to be. And then the last, acute transfusion reaction, remember these are talking about how fast they come up, right? The acute hemolytic transfusion reaction. These can be the intravascular or extravascular, depending on the mechanism of cytotoxicity that we're talking about. And these are the ones that are antibody-antigen reactions. So you're getting you've got antibodies to a donor red blood cell, or vice versa. You could be getting plasma and getting antibodies put into you that are contrary to your antigens. So in acute transfusion reactions, we said that they can be intravascular or extravascular. Some of the intravascular ones could be, the straightforward antibody-antigen reaction, but some of the extravascular ones can be things like the cells can be damaged before they ever get to you if they were not kept cooled before they were transfused. They can cause hemolysis extravascularly, and then you're giving people hemolyzed red blood cells, which is equally bad. Well, maybe not as bad as a incompatible cell, but it's pretty bad to get them. So those are the acute transfusion reactions of the type 2 hypersensitivity. Let's talk about the delayed transfusion reactions that were really considered. And these are an anamnestic response, so it's a secondary response usually to something that the patient was previously given. So generally, it's not going to be one of the major blood groups. You're not going to be transfused twice with the wrong major blood group, ABO, right? But you can be, as I said, there can be minor antigens on red blood cells that you get transfused with it the first time and you have a minor response, but it was a very low response. It was an IgM, it was a low response, but you've built a memory. And if you get that same kind of low dose or low frequency antigen, or kind of less immunogenic antigen the second time, your secondary response remembers it, your memory remembers it, and you have what's called a delayed hemolytic transfusion reaction. It's delayed because you have to have a period of time that you can produce some antibody to it. So you do have that delay until the IgG is produced. So the next of the major diseases of type 2 hypersensitivity reactions are hemolytic disease of the newborn. This again is going to be another antigen-antibody reaction, and it's caused by, we're going to see a picture on the next slide that will be helpful. But this is caused by a mother who is lacking a specific antigen, usually called the Rh factor. But now we know this term is a D antigen. When you're referring to Rh and D, in this case, we're talking about the same thing. You'll see there's a whole family of Rh, and they're different, but this is generally what we're talking about. It's just terminology, and it is, interestingly enough, when we're talking in blood bank about this blood antigen, we talk about it as a D antigen, but when we're talking about it in hemolytic disease of the newborn, we still call it Rh factor. So there you go. Anyway, I digress. Hemolytic disease of the newborn is caused by the development of antibodies by pregnant women who are lacking this antigen, but their fetus has it. So the mother is Rh negative, the baby is Rh positive, and what happens is the red blood cells of the Rh positive baby can enter into the mother's bloodstream either through delivery or even, just through during pregnancy. There can be minor passage of red blood cells. Without intervention, the mother develops memory and says, oh, I remember that red blood cell with that D antigen on it. I didn't know what the heck that was, and I built an antibody response to it, and can live perfectly fine because she doesn't have the antigen there. So the fact that she built an anti-Rh doesn't matter. She doesn't have it to begin with. But what is the problem is if she gets pregnant a second time and the next baby also is Rh positive, that's going to put that baby at high risk. And the mother will produce an anamnestic response. She'll produce IgG, which we know does what? It crosses the placenta. And so the IgG that the mother produces on the second exposure with the second child crosses the placenta, causes stillbirth, miscarriage, or even hemolytic disease of the newborn, where the IgG from the mother is going to attack the red blood cells of the baby and lyse them. Hemolytic disease of the newborn, you say, well, the baby's still alive. True, but it actually can be quite serious for the baby. Hemolytic disease of the newborn, the baby's red cells lyse. It releases hemoglobin. Hemoglobin breaks down and converts to bilirubin. And excess bilirubin buildup can cause brain damage. How do we get around this? It's actually quite simple. If a mother who is Rh negative is pregnant with an Rh positive baby, they can give her something called RhoGAM. It's an anti-D immunoglobulin or an anti-Rh immunoglobulin, and it binds on to the D antigen so that the mother can't produce any antibody to it. We're going to take a look at it. Let's look at the picture, and I think this will make sense. So we have mom. Mom is Rh negative. Baby is Rh positive. Baby's red blood cells leave the placenta either through birth or during pregnancy and encounter these naive B cells. Naive B cells, in the presence of this antigen, this Rh antigen, are induced to do what? To produce antibody, right? So in this case, because it's the first time they've seen this antigen, they produce what? IgM, right? The mother produces IgM. Can IgM cross the placenta? Why no? It's a very large molecule and it can't cross the placenta, so it's not going to go back and affect the baby at all. At this point, this IgM can't cross the placenta and get back to the baby. It's not going to hurt the baby. It's going to be in the mother. Mother doesn't have the Rh, so this antibody has no impact to her whatsoever. All well and good. Let's pretend the mother isn't treated. Now the same mother, still Rh negative, because she was born that way, has a baby that's Rh positive. This time again, there's a little bit of a bleed between the placenta and the mother. And we no longer have naive T cells, but we have B cells that are going to produce monoclonal antibodies. So it recognizes the antigen binding to this membrane-bound antibody, and these B cells are going to produce antibody. Because this is the second time the mom has been exposed to this, she's no longer just producing IgM, she's producing IgG as well. And we know that IgG crosses the placenta. This time the IgG goes back, crosses the placenta. It's going to sensitize the baby's red blood cells, causing red cell lysis, and I just told you the impacts of that. So it can be quite severe for the baby. As I said, there's a way to protect this. Either at the first pregnancy, in between the first and second pregnancy, right after birth. You do this right after birth. You can inject the mother with RhoGAM, which is an anti-D. So it's an anti-D or an anti-H. And you can see it comes in, it's injected in the form of an IgG, and it automatically binds off to the infant red blood cells that cross the placenta. And these naive B cells are never stimulated to produce antibody. Bing, bang, boom, done. Baby is safe, mom is safe, everyone's safe. Autoimmune hemolytic anemias are the next classification of type 2 hypersensitivity reactions. And this is a type of anemia that is directed against self-antigens. You produce antibody to yourself. It's autoimmune. You can produce two different kinds of antibodies, either warm reactive or cold reactive autoantibodies. Warm reactive do exactly what they say. They react at body temperature, 37 degrees. They're class IgG, and they're typically formed secondary to something else. You don't just all of a sudden develop autoimmune antibodies. You develop it after an infection like Mycoplasma pneumoniae or infectious mono, where they are heteroantigens in that they have antigenic epitopes that are identical to humans, and so you can produce antibody to them. These are also sometimes associated with things like B-cell lymphoproliferative disorders, where you're producing high volumes of B cells and thus antibodies. We can also see cold reactive autoimmune hemolytic anemias, and these are IgM in nature. They react at below 30 degrees centigrade. So since the human body is 37, it's insignificant for the most part. If they are in an area where it's cold, like your fingertips or your nose or your toes, or you're exposed to the cold, the antibody will bind to the red blood cells. And you know, IgM is a great little complex former, right? So you'll form lattices that can obstruct the capillaries and cause some pain and damage to the tissue there. Let's look at warm versus cold reactive antibodies quickly. So again, 37 for the warm, below 30 for the cold, IgG class, IgM class. While IgM reacts normally at below 30, and it binds to the antigen at 30 degrees, not minus 30, below 30, it will dissociate once it gets warm, once it gets to room temperature or body temperature again. So it's an interesting reaction. Warm reactive autoantibodies, no, I said that wrong. Warm reactive autoimmune antibodies can cause that hemolytic anemia that we're worried about as part of a type 2 hypersensitivity reaction, either extravascularly, where after you get antigen-antibody binding, phagocytes just love that and digest them and clear them out of solution, so extravascularly, or intravascularly by complement fixation. This is a lesser mechanism for hemolytic anemia with warm antibodies, because IgG is less effective at complement fixation. And as I said, it's usually secondary to infections. Think of infectious mono as a big one. It also can be secondary to other things like acetaminophen or penicillin, cephalosporins that absorb onto the red blood cell and induce an antibody production. They all of a sudden look foreign and they produce an antibody. They treat it with things like corticosteroids to suppress the immune system, and they'll actually give a splenectomy if someone has a chronic disease, because the spleen is constantly trying to clear these cells with antigen-antibody complexes on them. and you get increased symptomology of hypersensitivity with that. So cold-reactive antibodies, if we go down past the things we've already talked about, this is an IgM class, so it is capable of fixing complement, resulting in hemolytic anemia. Cold-reactive antibodies can be classified as transient or chronic. Transient ones are really just an antigen-antibody binding, forming a lattice, and when it gets cold in your fingers or your toes or your nose, or in very cold weather, where your body temperature drops, you end up getting obstruction in capillaries and some pain and some hemolysis there. Transient cold reactive antibodies, we also call those cold agglutinins. There's a disease called paroxysmal cold hemoglobinuria, where we're going to see in a second, where after a viral infection, patients produce an anti-red cell antibody. And we'll talk about it in a second, but we'll probably talk about it now and then get past it. This antibody is called biphasic. It's a biphasic cold reactive antibody. In that, it'll bind, see what it says, biphasic. It'll bind to a red cell at a cold temperature, but it doesn't get activated to hemolyze red cells until it gets to 37. So it needs both in order for this to become hemolytic. This is a pretty rare disease for a disease we talk about all the time. Let's see. Also, cold reactive antibodies can be a result of monoclonal antibodies that are produced in these kinds of B-cell dyscrasias, CLL, Hodgkin's, Waldenstrom's, all really strong B-cell diseases that produce cold agglutinins. And polyclonal, those are monoclonal because the cell lines are producing the antibody themselves. Polyclonal cold agglutinins we see after a couple of different disease states, things like mycoplasma or infectious mono, some viruses. So you can end up with cold agglutinins that are produced in a number of different ways. And so what's the treatment? So we have treatments here for warm reactive. We have some drugs and some interventions like a splenectomy. What's the treatment for cold? Avoid the cold. Just stay out of the cold and you'll be fine. And as I said, we'll spend 1 second on paroxysmal cold hemoglobinuria, which is an autoimmune hemolytic anemia of type 2 sensitivity type. It's post-viral, measles, mumps, chickenpox, infectious mono. I told you how it was biphasic. Causes intermittent hemolysis because it's biphasic. It's only activated at 37. So when it goes back out to the extremities, or you're no longer warm or cold, the hemolysis comes and goes. It's considered a disease of children and young adults, and it's, self-limiting and transient. Why? These are diseases typically of children and young adults, and the diseases themselves are self-limiting. So the paroxysmal cold hemoglobinuria, they can be post-virally stuff for the most part, and seen in kids. What kind of testing do we do for type 2 hypersensitivity? I think this is going to be our last discussion point for today, and we'll do the rest in class. Actually, I think I'll stop right here, and we'll do these in class on Thursday. So this is going to end Tuesday's lecture, and we will take this up on Thursday. Thanks. Bye.